Welcome to the Oxygen Alliance YouTube channel, where we bring you videos on installation, maintenance, and various trainings on oxygen equipment and devices. If you have comments and/or questions, drop them in the comment section below or write to us info@oxygenalliance.org. Remember to like our videos, subscribe to our channel, and hit the notification bell.
Hello everyone, thank you for joining us for this week's Oxygen Talk. My name is John Gozo and I'm currently in Lilongwe, Malawi. Today, I'm pleased to be hosting the call. Today I've been joined by our colleagues from OpenO2 and today they'll be discussing about pace oximetry. So in case you're just joining us for the first time, the Oxygen Talk is a video meeting that the Oxygen Alliance hosts every second and fourth Thursday of the month. And during this course, we have one call where our partners, they invite a guest and the guest discusses about a specific topic. And in the other meeting, we have our biomedical engineers from OpenO2 and Sunlight. They pick one topic and they discuss about it. And just a reminder for our new members, please do share with us your email address in the chat. You can just paste it in the chat, then we'll add you to our emailing list. We also have a Facebook page. Please do like and follow. We also have a YouTube channel. So please go ahead and check it out, subscribe, and you'll be able to see a lot of information that we have shared. Some of the recordings from the past weeks, they've been posted there. So without wasting much of your time, now we dive into our discussion. Over to you, Brenda.
Thank you, Sharon. My name is Brenda Luhanga, and I'll take you through pace oximetry. So this presentation is going to cover the background and justification of pace oximeters, their principle of operation, standards, test points and factors that affect their reading, and the common problems associated, but also their possible solutions. So pace oximetry is an invasive medical technology that is used to measure the oxygen level in patients' blood as well as their heart rate. This is an easy, painless measure of how well oxygen is being sent to parts of the body that are further from the heart. Pace oximetry has become an essential tool in healthcare settings worldwide, including Malawi, as it provides a quick and reliable way for assessing a patient's oxygen levels. This technology is particularly useful in diagnosing and monitoring conditions such as respiratory diseases, heart problems, and other oxygen conditions that affect oxygenation. Some of these conditions include heart attacks, heart failure, chronic obstructive pulmonary disease, anemia, lung cancer, asthma, pneumonia, just to mention a few. Pulse oximetry provides real-time feedback on patients' oxygenation status. This is a clip-like device called a probe that is placed on either the earlobe, the foot, or the finger. So before pulse oximeters were invented, several methods were used to analyze the blood concentration in patients. Some of the methods included clinical observations which involved in looking for signs such as labored breathing, cyanosis, which is bluish skin or mucous membrane discoloration, but also altered mental status. Arterial blood gas analysis was also conducted, which is a procedure that involves taking a blood sample from an artery to be analyzed in the laboratory for parameters such as oxygen partial pressure, carbon dioxide levels, the pH, but also bicarbonate levels. This procedure is, however, invasive and requires trained personnel and, of course, the presence of the laboratory. Continuing, the other method was transcutaneous oxygen monitoring, TCOM, which involves placing a probe on the patient's skin to measure the oxygen tension in the underlying tissues. This relied on the principle of oxygen diffusion through the skin. So Even though this achieved continuous monitoring of the patient's oxygen levels, it was usually affected by factors such as the skin perfusion, but also temperature. And lastly, the last method used was oxygen saturation estimation, where a healthcare provider would just estimate the patient's oxygen saturation based on the clinical signs, but also the supplemental oxygen delivery. So, for instance, they would observe a patient's response to oxygen therapy and adjust the flow rate accordingly. These methods presented a lot of limitations compared to pulse oximetry. So, some of the application of pulse oximetry, the information that is obtained from pulse oximetry is used in many kinds of situations, like, for example, during and after surgery, or any procedures that use sedation. It is also used to see how well lung medicines are working. Oxygen levels can also be used to check if a person's ability to handle increased, if a person has regained the ability to handle increased activity levels. It can also be used to see if a ventilator is needed to help with breathing or to see how well it is working during therapy. And it can also be used to see if a person has moments when breathing when treating sleep apnea. So pulse oximeters can be classified into three main groups based on their application, but also design sophistication. The first one is a finger clip oximeter. So this is a small and portable device that can be clipped onto a finger, which is usually the index finger as shown in the picture, and it provides an uninvasive, quick measurement of oxygen saturation. The second one is a handheld oximeter. So in this design, the pulse oximeter consists of a small unit with a display screen and a probe or a sensor that is attached to the finger, the earlobe, or the foot. So it is designed to be compact but also convenient for on-the-go use. The last class is a tabletop or a standalone pulse oximeter. So standalone pulse oximeters are designed to be placed on the table or any surface during use. They are usually larger in size compared to a portable handheld oximeter, and they often feature a steady base and a display screen that is easily readable from a distance, as shown on the picture. They are also capable of continuous monitoring and sometimes have enhanced functionalities such as alarming systems and also data analysis and data management systems. They can also come with connectivity and integration for transferring data to other healthcare systems or devices. These are usually powered with a direct power source, unlike the use of batteries, to ensure consistent power supply for a continuous operation. So this is all that I had. My colleague will continue with the principles of operation on pulse oximetry. Thank you.
Thank you very much, Brenda. Before your colleague proceeds, I just want to welcome those who are just joining us. Please, if this is your first time, to share with us your email addresses in the chat so that we can add you to our emailing list. And if you have any questions for us regarding this topic of discussion, please, you can paste it in the chat or you can just slide it down. At the end, we're going to have some time to ask the questions. Thank you. Over to you, Eliza.
Thank you, Sharon. My name is Elizabeth Tavalaba, and I will take you through the principles of operation on pulse oximetry. So there are two main types. One is transmittive and the other one is reflective. Starting with the transmittive, a probe is placed on a well-perfused area to shine light through the body tissue. This enables the assessment of the absorption property of oxygenated and deoxygenated blood to know the saturation levels. To the next type, we have the refractive. This one literally uses a sensor to detect the light reflected from skin surfaces, providing an alternating way to measure also the oxygen level. So as we can see from the figure to our far right, we're having two fingers placed on the LED and photodetectors. Starting with the one to our left, it is indicating the transmission operation. This one has an LED on top with a photodetector to the other end in opposite direction, while the reflection has like both LED and photodetector on the same surface. Continuing, but before we dive into these two principles, as biomedical technicians or engineers, we need to understand the technical aspects of pulse oximetry, which is so beneficial. So here are some of the technical details related to pulse oximetry. First, we have the light source. LEDs are used in pulse oximeters emit specific wavelengths of light. The red LED typically has a wavelength of around 660 nanometers, while the other is an infrared LED, which has a wavelength of around 940 nanometers. These wavelengths are chosen because they are within the absorption bands of oxygenated and deoxygenated hemoglobin. Continuing with our details, we have the photodetector. A photodetector is a transducer that converts the transmitted light into electrical signals. It typically utilizes a photodiode or a photodiode array, and this device generates a current proportional to the incident light intensity, which is then converted into voltage using a transimpedance amplifier. After that, we should also understand about the AC modulation. So to distinguish the pulsatile component of the blood from ambient light and other noise sources, transmittive pulse oximeter often use the AC modulation. So what happens is that the LEDs are modulated at a high frequency, typically around 50 to 100 kilohertz, and the photodetector filters out the ambient light by demodulating the received light signals at the same frequency. So as any other medical equipment, pulse oximeters needs also to be calibrated. This calibration ensures the accurate readings, which involves a comparison between the oximeter's measurements with the actual oxygen saturation levels obtained from the arterial blood samples. And after that, calibration curves or tables are created to convert the measured light intensities into oxygen saturation values. Then the signal is being processed using an advanced signal processing algorithm, whereby it extracts the oxygen saturation information from the detected light signals. These algorithms take into account factors such as the baseline, noise, motion artifacts, and variation in tissue characteristics. So signal averaging, adaptive filtering, and motion artifact rejection techniques are commonly used to enhance the accuracy and the ability of the measurements. Now, getting into our two main types of the pulse oximeters, let's start with understanding how the transmittive pulse oximetry works. As you can also see in this picture, we are having this pulse oximeter that actually has light through the LEDs and the photodetector in opposite directions. So just one can ask to say, really, what is a pulse oximeter? So it's like what we've seen here. Others have a probe and a display unit, only that this one has no probe. So what happens is that the probe is usually placed on a well-perfused area of the body such as the finger, as already mentioned, the earlobe or toe. And the two wavelengths of light, the infrared and the red, penetrate the skin and reach the blood vessels underneath. From there, oxygenated and deoxygenated blood carries oxygen to various tissues in the body. So oxygenated blood is, as we all know, is rich in oxygen. This absorbs more infrared light, which allows more red light to pass through. While on the other hand, deoxygenated blood has less oxygen. This also absorbs more red light and then allowing more infrared light to pass through. The emitted light then passes through the tissue and blood vessels in the body where the probe is placed, and some of the light is absorbed by the blood, while some is transmitted through to the other side since the photodetector is in the opposite direction. And in the photodetector, it detects the intensity of the transmitted light that reaches it. So it measures the amount of red and infrared light that pass through the tissue. After that, now there is signal processing. Here, what happens is that the photodetector converts the detected light intensities into electrical signals. And these signals are then processed by the pulse oximeter electronic circuitry. But we may understand that many times you can see a figure on the display. So there is some oxygen saturation that happens, calculations, and then they are now displayed and read out. So the calculated oxygen saturation level is presented on the oximeter screen as a percentage value. And this value represents the proportion of the oxygenated hemoglobin in the blood compared to the total amount of hemoglobin. And it's important to note that transmittive pulse oximetry provides an estimate of arterial oxygen saturation rather than a direct measurement. And it indirectly assesses the oxygen saturation level by measuring the light absorption property of the blood. Now, going ahead to the other main type, the refractive pulse oximetry. So this picture to our left is also showing that, whereby we can literally see that we are having both the LED light and the photodetector being on the same side. Actually, this is what really differentiates much between the transmittive and the refractive pulse oximetry. So a brief explanation of the process. The skin penetration, what happens is that when the LEDs are activated, they emit light that penetrates the skin and reaches the underlying blood vessels. So, and then after that, there is an interaction with the blood. As the light encounters the blood vessel, some of it is absorbed by the blood, while the rest is reflected back towards the skin surface. So in the refracted light detection, it also happens in a way that the information obtained about the properties of the oxygenated and the deoxygenated hemoglobin, it is then detected by the photodetector that then measures the intensity of the refracted light in the very same way, like the way I explained for the transmittive principle, whereby oxygenated blood absorbs more infrared light, allowing more red light to pass through, and also on the other side, oxygenated blood absorbing more red light, which allows the infrared light to pass through. So this is all that I had to explain on these two principles. I'll then extend this presentation to my colleague, Chimwemwe TSC. Thank you.
Thank you, Elizabeth and Sharon. So, going on the standards and requirements for pulse oximeters, up to today, the World Health Organization does not the World Health Organization had not released specific guidelines exclusively for pulse oximeters. However, the US Food and Drug Administration has produced a pre-market notification guidance So this guidance submission provide guidance on the design, performance, and testing of pulse oximeters to ensure their accuracy, reliability in measuring products in saturation levels. So this is intended for manufacturers, healthcare providers, and regulatory agencies to ensure that pulse oximeters are accurate, reliable, and safe to use in medical settings. So this guidance provides that one, a pulse oximeter should be tested under lab conditions for SPO2 accuracy. So this is mainly for the manufacturers before the product gets to the hospitals or to the market. So this falls under clause 201 and annex EE2 of the ISO 80601. B, the testing for SOP2 accuracy for oximeters should conform to clause 201 of ISO standard 80601. Another submission and guidance is that pulse oximeters should have visual and audible alarms as provided by clause 208 of ISO standard 80601. So you should make sure that when purchasing, or you should check in the user's manual that it conforms to this standard. So another standard is that the pulse oximeter should be safe with respect to electrical, mechanical, and environmental conditions. This falls under clause 201 and clause of the ISO standard 80601. The final standard, but this is not exhaustive of the standards that there are for the FDA, for the pulse oximeters, but we just took some. So the last one we compiled is that pulse oximeters should be compatible, biocompatible. This means that the condition of being biocompatible with living tissues or systems by not being toxic, injurious, or causing immunological rejection. This falls under ISO standard number 109. So it's also worth mentioning that individual countries may have varying standards with adopting the FDA provisions with little modifications to suit them. So it's important to always consult with local authorities. Thank you so much. My colleague will continue from here.
Thank you so much, Shimwe. My name is Grace Banari, and I'll take you through the testing points as well as the most limitation factors that affect the readings of pulse oximeters. So, beginning with, the testing points that are used mostly in infants are, there are several approaches. These are the finger approach, in-ear method, palm or so approach, forehead approach. The finger approach, this is the method, is the most common way to get your infant's pulse oximetry. When using this technique, the probe is advised to be placed in a correct position on the finger, making sure not to clamp the device too tightly, which would restrict circulation. And it should also not be loosely connected because this would drop or allow the light to enter. The other approach is the ear method, in-ear method. So pulse oximetry can be obtained through the ear access. With this technique, translucent probe clips onto infant's ear lobe, and it is connected to a monitoring unit that displays oxygen saturation levels. The third approach is the palm and saw approach. Mostly in newborns, a finger or the ear may still be too small to place the clip on, and the pulse oximeter may be attached to the infant's palm or foot. Finally, in infants, we also use the forehead approach. In rare cases where the fingers and the ears are out of reach, the pulse oximetry reflex probe can be placed low on the forehead and positioned just above the eyebrows to ensure placement away from the major blood vessels. When using this method, the infant should be reclined. In adults, reusable and disposable oximeter sensors should be applied to the ear lobe, finger, toe, bridge, or of the nose or the forehead. You should also note that each sensor is designated for a different part of the body. So the sensors are not used interchangeably. So a sensor for the finger or the toe should not be used for the ear or nose. It is also important to choose a pulse oximeter that is specifically designed for infants, as their fingers and toes are much smaller than those of adults. And additionally to this, it's important to follow the manufacturer's instructions carefully and ensure that the probe is securely attached to the infant's finger or toe. If you have any concerns or questions about using a pulse oximeter on your infants, it's the best way to do is to consult the healthcare professionals. And then I'll take you to the factors that affect the pulse oximeter reading. As already mentioned, we need to have a good supply of oxygen to the part that is being used to measure the pulse. So it is recommended that the area that is being used, there should be enough oxygen saturation in a way that there shouldn't be, for example, if you're using a finger to measure you're using the finger as a way of measuring, you should make sure that the finger is not cold, and this will help you to get the accurate measurements. And second to this is the increased movement. Increased movement during the testing period can also speed up your blood flow and make it harder to find an accurate measurement. For this reason, it is recommended that you sit in a resting position when taking your readings and avoid moving around while the device is running. And next is the skin pigmentation. So you should know that pulse oximetry works by beaming light through skin into the blood and inferring the oxygen saturation reading from the amount of the light absorbed. Therefore, higher levels of skin pigmentation could in theory affect how light is absorbed, thus possibly affecting the accuracy of pulse oximetry readings. Measurement inaccuracy could have a serious clinical implications, including the delay of urgent medical care. Yeah, so although some odd studies did not find any bias related to dark skin pigmentation, the latest studies have shown that pulse oximeters devices have some limitations with dark-skinned subjects, especially at a low saturation or hypoxemia conditions. Since hypoxemia is associated with increased mortality risk, there is a need to determine such errors exactly. Therefore, it is also worth to remark that to date, or up to date, the effect of skin pigment on pulse oximeter performed has been studied for only a relatively small number of pulse oximeter modules, and most of them have been calibrated using light-skinned individuals. To obtain reliable data to overpass the lack of features of the commercial oximeters on the market regarding dark skin pigment, more studies with other clinical and patient-reported data are required, and if possible, to redesign the device algorithm related to the light absorption with a calibration based on the dark skin patients. And another factor is the blood composition. So the oximeter can tell the difference between blood that is carrying oxygen and blood that is carrying carbon monoxide. So pulse oximetry may give falsely normal readings because it is not capable of distinguishing between the oxyhemoglobin and the carbon hemoglobin. Therefore, there's need to reduce the use of carbon monoxide containing substances such as the use of smokes. And next is the artificial nails and dark fingernail polish. So If you wear nail polish, especially the blue, green, or black or metallic ones, then the readings might be inaccurate because these materials can reflect the light back into the sensor while the device is still learning. And this stops it from fully penetrating the finger. And also, it can make it hard for the oximeter to be able to see the blood in the nose. Finally, I'll talk about ambient light as the factor that affects the readings of the pulse oximeters. So an external light, aside from the discrete wavelength employed by the instrument, can interfere with the measurement if it reaches the photo detector. Usually, ambient light pollution may cause offsets in the sensor or cause additional issues due to flickering of lamps at 50 Hz. That's introducing unwanted noise in the PPG signals, which can possibly lead to erroneous oxygen saturation values. For this reason, pulse oximeter sensors should be optically insulated from any external light, and they should be positioned carefully to avoid ambient light reaching the photodetector. On this far, I'm going to pass on to my colleague Boniface, who is going to take us through the common problems and their possible solutions. Over to you, Boniface.
Thank you so much, Grace. My name is Boniface, and I am going to take you through some of the common problems and the possible solutions that are associated with the use of pulse oximetry. First and foremost, for these devices to last long and to perform to their optimum best, we need to conduct maintenance. So in maintenance, we know we have preventive maintenance as well as corrective maintenance. So when conducting these maintenances, we engineers need to have some spare parts, and for most of the pulse oximeters, the spare parts that are required, which are associated with the breakdown of pulse oximeters, include the probes, the batteries, the LCD, the control PCB, as well as the buttons. So I'm going to take you through some of the preventive maintenance that we can carry out to make sure that our device have a long lifespan and do not have constant breakdowns. So first, you need to run a functionality test before applying to the patient. So this is just to make sure that the display and the probes are working in good conditions. You can try them when you're not using on any patient, on a normal person, to see if they are giving the right values. You also need to clean and disinfect the fingertip oximeter after each patient is tested according to the manufacturer's instructions and the infection prevention control protocol specific to the settings. So the cleaning is just to make sure you need to clean with at least 70% of alcohol. So this is to make sure that you avoid the transmission of germs and infections from one patient to another. And when it is not in use, you need to store in an appropriate place, which is well ventilated and secured, to make sure that you avoid children and other people who are not health personnel from interfering with them. You also make sure that you do not use expired or short lifespan batteries. Otherwise, that could affect the performance of whenever the battery is low. So you need to buy batteries that are long lasting. They don't have a short lifespan. This also reduces constant replacement that is required. So you don't want to be buying batteries here and there more oftenly. Now we are going to look at some of the problems that are encountered and the possible causes of such problems, as well as solutions that biomedical engineers and technicians can undertake to tackle such. So the first problem that we're going to look at is when you have a pulse oximeter that is not turning on. So there are scenarios where either you want to use the pulse oximeter, but it's not turning on. So the possible causes could be that, let's say, if you're using a standalone pulse oximeter, they have power cables and they are connected to a power source. So just make, it could be that there's no power from the main socket. It could also be that there's a fault with the power cable. And if you are using the handheld and the fingertip pulse oximeters, there could be problem with the batteries. It could be either that it is drained. It is also could be that it is discharged. As I've already said, that you need to use batteries that are longer lasting. So let's look at the possible solution that we can have. So first and foremost, whenever you're using a pulse oximeter, that is a stand on, make sure that there's a power on the main socket. So even though when there's power, you need to make sure that it is switched on. If that is not helping, you can also check the fuse to make sure that if it's not blown. If it is blown, then you can just replace it with a new one, which has the relevant, but also make sure that the current that you're using, the rating rather, it's compatible with what you had. You can also try the cable on another device. Just to, if it's working on another device, then that means there was another problem with the system, with the unit. But if it's not working, then there could be a problem with the cable. So you can run other tests like checking the continuity of the wires inside, just to be safe, to be sure rather. If you're using batteries, if you're using batteries, then just make sure that you change it. You can use a new battery. If you can just try to recharge it so that they work. Anyway, so let's go to another problem. Another problem that we encounter is having an unstable oxygen saturation or a pulse rate, or sometimes it's not displaying on the display unit. So it could be that the probe is not mounted correctly from the unit to the patient. It could also be that there's dirt on the probe itself. It could also happen that the patient is moving up and down, or that certain part that you have connected to is not still. It could also happen that the patient saturation is too low to be measured. Some patients could be anemic or have other disease that they have low blood in their system. So with low blood, it can't happen. You can also have a scenario where there's an internal malfunction. So that could also cause an unstable or not displaying anything. The possible solutions include make sure that you connect the probe and the cable correctly. So just check that the probe is connected to the unit and that is connected correctly. In certain cases, it could be that there's dirt, grease, or nail polish. So since you use different patients, there could happen that one had grease on them, and so you need to constantly clean it, like I've already said in the beginning. So the probe need to be cleaned often and remove if there's any dirt that is present. Make sure that you use alcohol. 70% of alcohol, that is. Also request the patient to remain still if they are moving up and down or they are unstable. For infants and neonates, try to locate the sensor on the foot or on any part that has already mentioned that is not moving up and down. You can also reset the probe if it is necessary. And if you have the device that has, let's say the PCB is you have problems with the PCB, then you may require that you replace either the PCB itself, or if it's a fingertip sort of type of pulse oximeter, then you can just replace it. So let's look at another problem. In other scenarios, you have an error display on the screen. So in most cases, it is usually a case where there is no enough voltage that is enough voltage that is being transmitted by the probe to the display unit. As a result, you cannot get a proper signal. So each and every brand has its own error codes. So sometimes there is error 1, error 2, error 3, and so on and so forth. So it just depends on the brand of the pulse oximeter that you are using. So the possible causes could be that there's a faulty probe, or there's a faulty control circuit inside the unit. So how can you deal with this situation? First and foremost, you need to replace the probe. If you have a spare one, just make sure that you use it and check if the problem is not persisting. If it is, then you can try to check the control board if there are no any other damages. And if there are damages, then just replace the control board. So they could be asking that why can't you just isolate a particular element on the control board and fix that? In most cases, if one element of the control board has malfunctioned, it happens that the entire control board should be replaced, because it's not normally that there's only one element that has malfunctioned. So it will take you time to actually isolate each and every component that is not working. So it is rather recommended that we should just replace the control board, the entire of it. Another problem that we should look at is when you have a discolored or black spots obstructing view display. So you could have a probe that is working, but the color on the display is it's really bad, or there are dark spots which could give you false readings, and that is not what we want. That could be fatal. So it's normally caused by a damaged LCD, because there are different type of medical personnel that use the pressure oximeter. So you can either be using too much force, and in a long run that could damage the LCD. So the possible solution for that is just to replace the LCD if you have one, and you make sure that if you have one, it is compatible with the brand that you are using. So make sure that you check the manufacturing, the manual guide, to make sure that you don't actually destroy whatever you had already. The last problem that is normally encountered is having a continuous alarm sounds. So the reason could be that you have alarm limits that are set too high or too low. So make sure that this could also cause the alarm to just be continuously making sounds. You could also have power that is disconnected. You can also have a scenario where there's an internal malfunction. So first and foremost, make sure that you set the appropriate time limit. Make sure that the power cable is connected properly, and if those have not changed anything, check the control board, see if you can fix it, and if you can't, then just replace the control board if you have a spare one. Having reached this far, I have exhausted what I have. Thank you for listening. Over to you, Sharon.
Thank you very much, Boniface, for that explanation. Now it's time for the questions. So if you have any question, you can just raise your hand or you can paste it in the chat. So we had a hand up from Henry Milazi. Henry Milazi, if you still have a question, please you can come off mute and ask your question. Henry, are you still with us? Okay, nothing from Henry. Okay. All right, we have a hand from Raphael. Please go ahead.
Thank you very much for the wonderful presentation. And yeah, it was really interesting. My comment is it would have been again nice for the users to listen for these presentations, the doctors, nurses, clinicians, because some of the standards are violated by us, the way we use. And again, we don't know how to tell the engineers. After we have tested all the oximeters, we need again to recommend to you to say this is the nice brand to you. I don't think this is happening because people, they just receive any brands they are using. So it would be nice again sometimes we have a chance so that at least these people should be around. I didn't hear about anything concerning the failure rate, because as you are saying on the standards, when we say it should pass quality control, it means there should be some failure rate. And I think these ones, I didn't hear anything. And again, on replacing the probe and other things, at the end, when the machine is not in use, next, I would have again be interested, and not now, how we dispose these machines in case we cannot even repair anymore. Do we just burn? Do we just maybe heap in a bin? How do we dispose them? Would have been very much interested if I had those information. Thank you so much.
Thank you very much, Rafa. Okay, Timmy, do you have anything to say about that? Our fellow engineers.
Thank you, Sharon.
Okay, go
Thank you.
ahead, go ahead.
Yeah, so currently we don't have information concerning the lead of area about the pulse oximeters. I'm not sure. I don't know if anyone has an answer to that and can come off mute and present.
Yeah, if we don't have, it's just something to be thinking around, so that at least, like myself, when I'm using a blood pressure machine, I prefer a manual one, because the digit, again, I'm afraid of those failure rate. And when I'm using the pulse oximeter, again, if I'm having some problems, again, you have to, as a clinician, to learn how to rate the coma scale. For the children, maybe out of 10, for the adults out of 15. That will just beef up on any problems, on any information, if you have the problems with the machine. So if we don't have now, it's okay, but maybe as young guys, we can be doing even some research to say a good one after passing the test, maybe the failure rate should be maybe 0 point something percent, or maybe 1%, 99% okay, so that at least people cannot use, people can use within that limit. I was just thinking around anyway.
Thank you very much, Rafael. I think it's a good thing. It's something that we can look into and have and gather some information about it. And maybe someday we'll share about it. Thank you very much. We have a question in the chat, and it reads, how would you calibrate a pulse oximeter? What are the steps? How would you calibrate a pulse oximeter? What are the steps? So over to you, our engineers. Anyone who can help a colleague? Yes, Kelvin, over to you.
Thank you, Sharon. So I believe as Elizabeth was presenting, she mentioned a bit about calibration. So during manufacturing, the pulse oximeters are calibrated based on oxygen levels of lab samples of blood. So you take a sample of blood with known concentration, and then you start comparing that value with the one you're getting from your machine until you get to the accurate reading or thereabout with some given tolerances. But as for a pulse oximeter that you have purchased and you want to calibrate, unless there's a setting in that machine that requires calibration, I do not believe that we have a way of calibrating it. So when you're calibrating, you compare the measurement accuracy of a machine to a non-standard. So if it has that setting, you might just be, you can just compare that that accuracy with a pulse oximeter that already exists, maybe in your hospital, which you are confident that works. Yeah, that's my take on how to calibrate pulse oximeters. I hope that helps.
Thank you very much, Kelvin. We also have a hand from Grace. Go ahead, Grace.
Okay, I think there was also another question about how to dispose the pulse oximeters. So The best way of dealing with this issue is to check with the manufacturer if they have some sort of recycling program when it comes to the device. Especially the way this works is that these manufacturers have certain programs that incorporate special disposal method in order to have a little impact on the environment as possible. Thank you.
Thank you very much, Chris. Do we have any further questions or comment from the audience? Okay, it seems we have no further questions. So looking at our time, is almost up. So I'd like to thank you all for joining us for this week's Oxygen Talk. So please join us again next time when we'll be meeting. So for those our new members, in case this is your first time, everyone, please do share with us your email addresses in the chat so that we can add you to our emailing list. So we'll be sending you a calendar invite showing when we'll be meeting again and what we'll be discussing about. So please share with us your email addresses. Also, if you have any questions for us regarding maybe today's topic or any other previous topics, or maybe any other question about concentrators or oxygen related equipment, please don't hesitate. You can send it to us through our email info@oxygenalliance.org, and we'll do our best to give you the right answers. We also have a Facebook page. Please check it out. You can like our page. You find a lot of things there. We also have a YouTube channel. So we have the recorded of all past chats. You find them there. We also have videos of some maintaining oxygen concentrators of different models. You'll find everything there. So please check it out and don't forget to hit the notifications bell. So until next time is goodbye from me. Bye.
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 Bye bye.